Okay, student, Assalamu alaikum and welcome back to the classes. Although these are online, but we have to take these classes. So today we will discuss about the cancer vaccines. Last time we were discussing this lecture from the paper of Turning the Corner on Therapeutic Cancer Vaccines. I think most of the portion of this lecture have been covered, but I am going to start it from very beginning so that we may cover some other points which are very interesting and important for this lecture. So before the lecture starts, we have to see some of the figures which are actually representing the immune responses, the behavior of the T cells and B cells, and what are the roles of the T cells in initiating the overall adaptive immune response. So let's see. Here we can see the first figure, and according to this figure we are seeing the generation of some T cells and B cells along with the cytokines. Here you can see the important cytokines. Cytokines are very important for the development and duration of the development of the immune system. So here you can see that these are two cells which are very well-known cells, T cells and B cells. So how the T cells are going to respond towards the B cells? So here helper CD4 cells and cytotoxic CD8 cells, these are produced by the T cells. These are two types of T cells. And when we say that the CD4 cells, so we are talking actually about these helper cells. And similarly, CD8 cells are cytotoxic. Here you can see the cytotoxic T cells. And what are the functions of the cytotoxic T cells? So they activate the cytotoxic cells, which alternatively kill the virus infected cells. On the other hand, if we look at the helper CD4 cells, then they produce two or three cytokines, which are known as interleukins, and we denote IL as interleukin, which are the messengers of immune system, and different cells transfer their messages to another cells by the cytokines, and these cytokines either activate the cells or deactivate the cells. Here you can see helper CD4 cells produce IL-2, and IL-2 is not only required by the CD8 cells so that these cytotoxic cells can be activated. On the other hand, IL-2 is also activating the helper cells and macrophages. Macrophages are those cells which are actually engulfing all of the antigens. So activated helper cells have been activated by the presence of IL-2, and in response inhibit intracellular bacteria and fungi, whereas these helper T cells have a specific function to destroy the bacteria and fungi, whereas cytotoxic T cells have a specific function of virus infected cells and kill the virus infected cells. So this portion, after this portion, we can see here that the IL-4 and IL-5 two cytokines are very important to be considered in the production of the plasma cells by the B cells, and these plasma cells alternatively or in the long run produce antibodies, and these antibodies not only neutralize the toxins and viruses but also with the complement and neutrophils can respond back towards the bacteria. This is another figure in which we can see the presence of the APCs and virus immunogens. So what is the overall workflow? And let's discuss it first. So virus immunogen have been taken by the APCs, and APCs are antigen presenting cells. For example, macrophages or dendritic cells. After the presentation of the APCs, the class 2 MHC proteins have been flagged out on the APCs, which is basically flagging the portion of the virus, these class 2 MHC proteins. And a viral epitope, as you can see here, this has been flagged by the MHC, and this viral epitope is then presented to the helper T cells. So here are the MHCs with the viruses, and these are interacting with the TCRs. These are the TCR receptors, whereas this is the MHC plus virus, or not the complete portion, but the small portion of the virus. So these helper T cells are expressing another receptor cell on the cell surface, which is CD4. And here you can see the CD4. These helper T cells are also producing the IL-2 receptor, as we have discussed in the previous figure. And these helper T cells, basically, which are the initiators of the B cells, memory Th, or helper T cells, and cytotoxic T cells. So helper T cells are inducing IL-2, and by this these can be differentiated into the CD cytotoxic T cells, which are also known as CD8 T cells. So these cytotoxic T cells actually induce cytotoxins, and which are very important to kill the virus in the long run. And here these cytotoxic T cells are also differentiated into the memory TC cells, memory TC cells. When these virus again attack the system, then these memory T cells are very important at that time to respond it back earlier, because the first response is a little bit slower, whereas the second response, when the memory TC cells are present, then this response takes lesser time. Similarly, here you can see the memory TH cells, which are also produced during this response, immune response. And this is very important. This side overall is very important, in which we can see that antibodies are going to be formed, and these antibodies, along with the memory B cells, are also required for the earlier induction, basically. In thymus and in bone marrow of the human beings, which is Burza equivalent, these are produced, and in thymus you can see that cytotoxic T cells, or CD8 positive cells, and this CD8, remember the 8 with the cytotoxicity, cytotoxic response, or T cells. So after thymus, you can also see here that the helper T cells are also present, which are known as the CD4 positive. And by the presence of helper T cell, actually, these cytokines, or interleukins, are produced, and these interleukins are very important for the maturation of cytotoxic T cells and the B cells. Here you can see the differences of or comparison of the T cells and B cells. Antigen receptors on surface, either these are present or not. Then T cells and B cells both carry the antigen receptors on the surface. IgM on the surface is present or not. Then B cells, in the B cells, IgM is present. IgM is antibody, and antibodies are of two types, which are actually present in the system as a soluble form, which are known as immunoglobulins, and whereas IgM on the surface is the receptor. Here you can see another thing, which is IL-2 and IL-5, 4 and 5 with the gamma interferon synthesis. This is only present in the T cells, and B cells require them from the T cells. So T cells will actually give the IL-4 and 5 for the maturation of B cells. Long story short, these are all of the same figures, repeated again and again in one way or another. And actually, these are representing the specificities of the overall immune response between the T cells, B cells, MHC, CD8, CD4, all of these things. So here is a table in which type of cells, helper T cells means CD4 T cells, and cytotoxic T cells means CD8 T cells are recognized by the representative receptors on the surface of the cells. These are surface proteins, which are CD4 for the helper T cells, CD8 for the cytotoxic T cells. In the B cells, we can see the IgM is present, which is not present in the helper T cells and cytotoxic T cells, as we have studied or as we have seen in the previous figure. On the macrophages, class 2 MHC is present. It is very important to be remembered that class 2 MHC is most of the time is present on the antigen presenting cells or the macrophages. Here, macrophages plus APCs can be taken where the class 2 MHC present. This class 2 MHC plus CD4 or CD8 is very important to be remembered in all of the immune systems and all of the immunological studies. Natural killer cells, NK cells, these are also known as NK cells. This has the receptors for class 1 MHC, and this class 1 MHC receptors are mostly being represented on the natural killer cells. All cells other than mature red cells have class 1 MHC. So this is the presence ratio or the presence of the of different surface proteins for which they are known. Main functions of helper T cells, IL-4 and 5 is activating B cells, whereas IL-2 is activating cytotoxic T cells and activates the antigen-specific helper T cells to produce a clone of these cells. Clone of these cells means that helper T cells are going to produce the same information in the progenies. It has the same information in all of the progenies. So this is the cloning. I will attach all of these figures, and along with the book I will send you the lecture. So there will be the three things: book, these figures, and lecture. Comparison of Th1 cells and Th2 cells. This Th1 produces IL-2 and gamma interferon, which is the Th1 cells, whereas Th2 cells, they cannot produce IL-2. So what is the story of Th1 and Th2 and where they are coming from? Basically, naive CD4 T cells. Naive means they have not been contacted so far with the antigens, and these naive T cells are differentiated into Th1, Th2, Th17 and Treg cells. These are four classes which have been studied so far. And Th1 and Th2 perform different functions in regarding the excretion of the cytokines, where Th1 cells secrete IL-2 and Th2 cells secrete the major portion of IL-4 and IL-5s. So the signature cytokine of Th1, Th2, Th17, and there are a lot of Th cells based on the interleukins which are going to be produced by these cells. So gamma interferon is for Th1 cells, interleukin 4 are for the Th2 cells, and interleukin 17 is representing Th17 cells. So these cytokines are basically the proteins which are actually the messengers, and these messenger proteins are moved from one cell to another so that the message can be conveyed from one cell to another, that whether you are going to be matured or not, or you should be activated or not. So these cytokines have this kind of response in immune responses. So antigen presenting cells carrying bacteria, virus, cancer immunogen, or anything like that which is external or alien to the body, and it is non-self protein or non-self epitope or non-self bacteria or something. So antigen presenting cells are going to present it, but antigen presenting cells must have class 2 MHC protein. This class 2 MHC protein is presenting the antigen, and the antigen class 2 MHC protein plus TCRs are actually required for the generation of helper T cells. And in the long run, helper T cells then again produce IL-2 or interleukin 2. These helper T cells with the TCRs are or CD4 proteins. Here you can see the CD4 protein, which should be present along with the TCRs so that it can have a signal from class 2 MHC protein, antigen, and from the APCs. All of these things are actually required so that helper T cells can initiate the overall response. On the other hand, if the virus infected cell is producing some flag on the surface, then it is taken up by the class 1 MHC protein, for which the CD8 protein and TCR are also required, so that cytotoxic T cells should come in the way for the immune response.